Hello, everyone. I welcome the students, researchers, scholars, and scientists around the world on Advanced Multiple YouTube channel. Advanced Multiple actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Okay, well, I'm going to share my knowledge, my experience, and my research under the title of Application of Polymerase Chain Reaction. For this purpose, I collected the information and scientific data from different sources like PubMed, Medline, Scoopers, Google Scholar, National Library of Medicine, MBAS, directly of Open Access Journal, DWAJ, ProQuest, CNKI, that is China-based database company, stand for China National Knowledge Infrastructure. Some of the information collected from ResearchGate and Crossref. Okay, this is the photograph of Mr. Dr. Professor Carey Mullis. In 1985, he actually invented a new precise method to select and amplify a selected piece of the DNA or DNA component. Dr. Carey actually received a Nobel Prize in Chemistry in 1993 for his invention of polymerase chain reaction. This is the Dr. Carey Mullis. Before moving forward, I would like to talk a little bit about introduction and component of PCR. PCR is a relatively simple and inexpensive tool that can be used to focus in a segment of DNA and copy it over million of times. PCR is used every day for the diagnostic purpose, identify bacteria or the viruses, match criminals to crime scene, and many other scientific purposes. Okay, well, before discovery of PCR during the mid 1980s, the only way to make many copies of the DNA was to insert the DNA pieces into the bacteria and select the desired one from many different colonies growing on a plate. That was manual method to identify and clone the DNA segment. The component involved in PCR are 8. These are DNA template, primers, DNA polymerase enzyme, nucleotides, and buffer solutions, divalent ions, distilled water, and thermal cycler. Okay, well, the real-time PCR versus traditional PCR, we are going to compare these two different models. Real-time PCR allow for the detection of the PCR product during the early phase of the reaction. This ability of measuring the reaction kinetic in the early phase of PCR provide a distinct advantage over the traditional PCR detection. And the traditional method use gel electrophoresis for the detection of the PCR amplification in final phase or at the end point of the PCR reaction. Okay, well, this is very simple linear graph that show and demonstrate or highlight the basic fundamental thermal cycler profile. So if we call linear graph, it means it should have technically two parameters, horizontal and vertical. Horizontally, we took the time in minute, and vertically, we take the temperature in degree centigrade. We have 3 steps: step 1, step 2, and step 3. These are the basic fundamental steps involved in PCR thermal cycling. Okay, well, in step 1, there is a temperature of approximately 95 degrees centigrade maintained for about 1 minute or 60 seconds, that called as heat denaturation. At this phase, the DNA strand is split into the two chain. Second step is called as primer annealing. The temperature of about 75 degrees centigrade is maintained for approximately 40 to 50 seconds. And the third step, that called as synthesis of complementary chain, it comprises of approximately 50-second temperature maintained about 94 degrees centigrade. And this is actually fundamental basic profile or introduction of the operation of PCR machine. Okay, well, this is another diagram. It actually highlights the polymerase chain reaction, all 3 steps involved in its whole operation. There are the 3 steps involved in PCR machine: step 1, step 2, and step 3. In step 1, it called as denaturing or the split of the DNA into the 2 strands, template strand and other strand, complementary strand. Okay, well, in step two, there are three components involved: DNA primers and DNA polymerase, enzyme, and nucleotide. We know the nucleotide are four main types, that is adenine, guanine, cytosine, and thymine. They are the building blocks of the DNA. At the third phase, the building blocks, they join together, and the DNA polymerase enzyme work to, to build and put the nucleotide at their exact position, and a DNA strand or synthesis successfully. So this cycle is repeated for about 20 to 40 times to produce or synthesize the clone of particular DNA segment. Okay, well, what is the scope of the PCR? Real-time PCR scope having the six different points or six different scope of the PCR. First one is the quantitative messenger RNA expression study. The second is DNA copy number measurement in genomic or viral DNA. Next one is allelic discrimination assay on SNP genotyping. Verification of the microarray result. Then fifth one, drug therapy efficacy, and sixth one is the DNA damage measurement. Okay, well, what is the application of the PCR? There are the seven different application of PCR used sufficiently in market for the academic research and clinical purpose. PCR technology facilitate the detection of the DNA or the RNA of pathogenic organism in clinical practice. A variety of infectious agents are isolated and identified in diagnosis, progression, and therapeutic purpose. So PCR-based tests have numerous advantages to determine the body immune response. Most common use of the PCR are determination of pathogen and pathogenicity. PCR-based diagnostic test, diagnosis of genetic diseases, genetic counseling. The fifth one is forensic science application, then application in research in molecular biology. The last one is the other miscellaneous application. Okay, well, now we are going to discuss one by one determination of pathogenic and pathogenicity. In particular, PCR-based tests are competent to detect the presence of pathogenic agent in advance. Then serological-based method, as patient can take a week to develop antibody against particular contagious infectious agents. So PCR-based tests have been developed to enumerate the amount of the virus in a person's blood, like viral load, thereby allowing physician to check the patient disease progression and response to therapy or the treatment or medication. This has incredible potential for improving the clinical management of the diseases caused by viral infection, including AIDS, hepatitis, assessment of the viral load throughout and after therapy. Okay, well, PCR-based diagnostic tests are available for detecting and or quantifying a number of pathogens, including HIV, which cause AIDS, hepatitis B and C viruses, leading to viral cancer, human papillomavirus cause cervical cancer, chlamydia, trachomatis lead to infertility in women, Neisseria gonorrhea lead to pelvic inflammatory disease in women, cytomegalovirus that cause life-threatening disease in transplant patients, and other immunocompromised people, including HIV and AIDS. Mycobacterium tuberculosis, which in its active state cause tuberculosis and can damage the tissue of the lungs, kidney, and bones. Okay, well, the next application of PCR is the diagnosis of genetic disorder. The use of PCR in diagnostic genetic diseases, whether due to innate genetic change or as a result of natural genetic mutation, is becoming more common. Abnormality can be diagnostic even prior to the birth of the baby. Single strand conformational polymorphism SSCP or single strand chain polymorphism is defined as conformational difference of a single strand nucleotide sequence of identical length as induced by differences in sequence under certain experimental conditions. These days, SSCP is most applicable in diagnostic tool. In molecular biology, it can be used in genotyping to detect homozygous individual of different allelic state, as well as heterozygous individual who inherit genetic aberration. Okay, well, genetic counseling. The usage of PCR in genetic counseling is done for the parent to check the account of genetic disease beforehand to make a decision of having a baby. Detection of genetic disease before implantation of the embryo in IVF, it means in vitro fertilization, also known as pre-implantation diagnosis, can also be done exploiting PCR-based method. The diagnosis of inherited or spontaneous disease, either symptomatic or asymptomatic because of family history like Duchenne muscular dystrophy, PCR-based method is very useful. Okay, well, in forensic purpose, PCR used for genetic fingerprint is one of the most exploiting application of the PCR known as DNA profiling. Profiles of the specific stretch of DNA are used in genetic fingerprinting. Generally, 13 loci are compared, which is differ from person to person. Okay, PCR also play a vital role in analysis of genomic and mitochondrial DNA analysis, in which investigation use samples for hair, shaft, and bones when other samples are not accessible or available. Okay, well, utilization of PCR in research in molecular biology. PCR is an essential technique in cloning procedure which allow generation of large amount of pure DNA from tiny amount of template strain and the further study of particular gene. Some alteration to the PCR protocol can generate mutation, journal or site directed in a sequence either by an inserted DNA fragment or base alteration. PCR is used for the sequence tracked site (STS) as an indicator that particular segment of genome present in a particular clone. The common application of the real-time PCR is the study of expression pattern of gene during different development stages. PCR can also investigate on and off of a particular gene at different stages in tissues or even in individual cells. Okay, well, there is also certain miscellaneous other application of the PCR, including PCR has numerous application in various fields. The Human Genome Project for determination of the sequence of a 3 million base pair in the human genome rely heavily on PCR. The gene associated with a variety of disease have been identified using PCR. PCR can help to study DNA from various organisms such as viruses, tissue, bacteria, protozoa, yeast, fungi. Then PCR have been used to identify and explore relationship among species in the field of evolutionary biology. In anthropology, it also used to understand the ancient human migration pattern. In archaeology, it has been used to spot the ancient human race. PCR commonly used in paleontology to amplify DNA from an extinct species of crypto-reserve fossils of million years, and thus can be further studied to elucidate currently, like in present date. So, in conclusion, PCR is an effective tool of fluorometric detection, real-time measurement of initial concentration of targeted DNA. It has a vast dynamic range with high degree of sensitivity. So, this powerful machine and technique is reliable and reproducible. However, it can potentially be used in clinical, forensic, food research, technical, and pharmaceutical science to cope with the post challenges. Okay, well, before closing, I would like to salutate and greet to my colleague, my team, my members, Sherry Bajkar, Wolfgang, Daniel, Dustin Fakar, Marwa Al Maghribi, Terry Mill, Faisal, Noreen, Hamza, Robert Heyman, Amin, Arslan, and Ishtiaq. I also acknowledge the contribution and the kind support of Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Thank you very much for staying with me and watching the video. See you soon in the next video. Take care and bye for now.